I'm Allison Johnjack, the Cranberry Outreach Specialist with the University of Wisconsin-Madison Division of Extension. These videos are the on-demand recordings of UW Extension and the Wisconsin State Cranberry Growers Association's virtual brown bag lunch series. These monthly sessions offer growers and researchers a way to share timely and relevant information. We hope you'll enjoy and continue to engage with us.
Hi, I'm Tom Lochner, the Executive Director of the Wisconsin State Cranberry Growers Association. We're excited to be a partner with Extension to bring you research-backed resources, tools, and tips to keep Wisconsin marshes productive and profitable.
Thank you. Welcome to the June edition of our virtual Cranberry Brown Bag. We're going to get to go through a little bit of time-sensitive and on-the-spot information, both from researchers as well as from some of our product suppliers, research and scientists in the field. You have closed captions down on the screen below you, and if you're not seeing those, you should be able to hit the little ... ellipsis to say show more and show captions. I think that's it for the housekeeping, apart from have yourself on mute unless you want to ask a question. We do encourage people to ask questions throughout, and you can either use the chat if you want to ask a question publicly, or you can chat and use the little drop down to Allison Johnjack, and then I will ask your question on your behalf anonymously. The other thing you can do is unmute yourself and ask directly. And I think with that, usually I would hand the controls over to Tom for a minute. Tom Lochner of the Wisconsin State Cranberry Growers Association. He is on the road serving growers today, and as you can kind of see, we're all kind of on the road today. But Tom wanted to thank people who made it to the golf outing fundraiser that was on Wednesday. And, we've got a good lineup today. Dave Jones, the senior agricultural scientist at Ocean Spray at the Babcock station, is going to be giving us some in-season observations. Then Christelle Guedot, whose E apparently accidentally turned into a Polish E instead of a French E. I apologize for that, Christelle. But still, Christelle Guedot and Olivia Lake will be talking about, from Christelle's behalf, the current nematode research project that's going on, and then from Olivia, information on the nematodes that are available from Copert. And then in between those two, I'm going to have a quick review of new information in the chemistry world as we're having product availability, new updates, and then just a reminder to go ahead and ask questions all along and as we go.
I was asked to give a quick overview of the nematode trials that we're conducting this year. So disclaimer to start off, I do not have any data. So please don't expect any data because this is ongoing. This is a project that's being run by my lab in collaboration with Morgan Weisner, who was the student doing this project and already in the field working with growers, and also in collaboration with several cranberry growers and Dave Jones and Sean Steffen. So commercial nematodes against flea beetle. So I'll start by giving you, if I find my key, there we go. So this is really targeting red-headed flea beetle. As we all know, with the flea beetle, they cause that skeletonization, can have the leaves turn red, prematurely red. And so there's the concern about what that will cause in the next year production, but also we know that flea beetle can cause surface scaring on the fruit, surface feeding, and can potentially cause fruit feeding damage, but primarily it's on the leaves. And as we know, and I'll talk a little bit more about that in a future brown bag, on what we know about the life cycle of flea beetle, is that they overwinter as eggs here, which in Wisconsin, with the populations we've tested, they will require as eggs about 15 weeks at 5 degrees Celsius to overwinter. And then after that, they will take about 3 weeks to hatch. So they spend the bulk of their time as eggs. On the larvae, when we try to have a colony in the lab, and it's really hard, I'm talking with a lot of colleagues and nobody has had a colony of flea beetle. But in the lab, what we've seen is about 3 weeks as larvae, But we don't know exactly because we only got one pupa and then nothing else survived. So it's really hard to keep them going, probably due to the moisture that they require for that winter period. What we did is we went in the field and did soil cores, and we presented that data a while ago, but I just wanted to show you that again, and I'll talk about it again in the future this season. But we see, as you can see on the y-axis, that's the date, and then this is the total number. So it's not an average number per week or anything like that. It's the total number of eggs in black and larvae in that dark gray. And so we didn't see anything in those soil cores and before June 19th, where we saw a lot of eggs, and then I started seeing some larvae. And we see the larvae until early August. And after that, all we saw is eggs again. So probably the larvae and then all the eggs that are being laid, that will be the next batch of eggs that will overwinter. So we're thinking, but nothing is set in stone. We still have to do more work on this. This is work that was done by my postdoc, Ben Jaffe. But it spent about 8 weeks for all the way from the larvae that will turn into adults, they lay eggs. It's about that time, but nothing is set in stone on there. But as you can see, we didn't see anything in May, June, though we know there are eggs at that time. We just couldn't find them. So they're very, as we know, patchy and hard to find in there. What we also looked at is where they were laid in the depth of the bed. And so what we found is that we have a lot more eggs at the 15 to 30 centimeter depth than the 0 to 15 centimeters. So what that means is that the female are digging down into the ground to go lay those eggs really deep, at least at 15 centimeters. Down. It could be anywhere. It could be at 16 centimeters. We couldn't, with those soil cores, be very specific, but there's more further down in the ground. And that is important when we think about nematodes as a potential target for those, or not target, but potential killing agent for those flea beetle. So the nematodes, we all know why we're interested in them, but I just wanted to go a little bit backwards and provide a little bit of information about them. So they're those round worms, unsegmented. They're very abundant in most soil. They're on every continent except Antarctica, and mainly because there's only 3 species of insects in Antarctica. So that explains why there's not really that many, really no entomopathogenic nematodes there. There are nematodes in Antarctica, but no entomopathogenic nematodes. What's nice about them, besides the fact that there's no human toxicity and all of that, is that they cause mortality very quickly of the host, within 48 to 72 hours after infection. So they can die very quickly. And they are used primarily on soil-dwelling insects, but they have been used on other insects that are not soil-dwelling. One of the work is the nematodes that Sean Stephan has been working with can kill spotted ring Drosophila, which spend very little in the soil, on the soil surface, but it's probably also on the foliar spray acting on the larvae there. So not soil-dwelling insects necessarily. Looking at the life cycle, I'm sorry. If we start when the entomopathogenic nematodes, so EPN is what I will say from now on. When the EPNs enter the host, they enter through natural orifices, such as the mouth or the anus or places like that, or through the cuticle. And they enter there, and they will have 4 juvenile stages. And the 3rd juvenile stage is the important one. It's called the infective juvenile, and then the adults. So they are always associated with symbiotic bacterium that they have ingested, and that after they cut the lining of the guts, they will release those bacteria into the hemolymph of the host. And so the host will die because of the toxins that the bacteria are producing via septicemia. So the bacteria are there, they're killing the host. Inside the nematode, there is reproduction. Either there's mating and there's regular reproduction, or it can be parthenogenesis. and they will be reproducing within the host. And there can be 2-3 generations within the host before they deplete all the nutrients within that host. And then they will stop that stage and stay at that infective juvenile stage, the third stage of the juveniles. And they will then fill up the host and then be released into the environment as infective juveniles that will go and find another host. So there's a lot of considerations, and you've heard about them, but they are very sensitive to temperature extremes, very cold, very hot. Drought is also a problem because they need the moisture into the soil to survive. They're sensitive to solar and UV radiation, to desiccation. They require a pre- and post-treatment irrigation for the most part. It's very important to water them in because, again, they're very sensitive to desiccation. They're largely incompatible with insecticides, but that's not entirely true. So there's a lot of research that needs to happen with that. But for the most part, when you're applying them, you wouldn't want to be applying an insecticide, and you wouldn't want to tank mix nematodes with an insecticide, even though they're not insects. They have a limited shelf life and availability, but there's a lot more work that's been going on, and that's where Olivia will come in and talk to us about that. But we're able to keep them, and we're able to get them as well. But it's something to keep in mind. And then the most important is the handling and application technology that are really essential to the success of applying nematodes for controlling pest insects. So when we're looking at those nematode species, we have really the main genera that we have are steinernema, Neo-steinernema, and there's about 61 species in there, and heterorhabditis with 16 species in that genus. Each of those different genera, steinernema and heterorhabditis, are associated with different bacterium. And you have the names there, Xenorhabdis or Photorhabdis bacterium. We don't know, there's not enough research on nematodes, and especially on the bacteria that are symbiotic within the nematodes. Exactly how virulent each bacteria is, how they're really going to be acting on those different insect species. But they only survive within those nematodes. So they are not going to be a free-living bacteria. They're completely associated with those nematodes. And also what we don't know is the bacterial payload. So what you would call for bacteria is the number of colony-forming units. And the number of those colony-forming units per nematode species varies. It varies per the bacteria species or bacterium species, but it also varies per the nematode species potentially. So that's also something we don't know a lot about. And the more bacteria you have, the more virulent it could be, but not always necessarily, because it goes back to how virulent is the bacterium in the first place. So I'm just telling you that there's really a lot we don't know pretty much on how that all works and how the virulence is really happening. There's two main strategies for foraging, and those are important when you're thinking about a specific insect pest. So they can be ambushing or cruising nematodes. So when they're ambushing, they wait at the surface of the soil for the host to come close to them. And that's the main case for steinernema species. That's what they do. So they will wait for the host to come close. So if you have an insect that's not really mobile at any stage, whether it's at the larval stage in this case, or it could be at the adult stage, then that's not going to be the best option here. For the cruising nematodes, they cruise through the soil, and that's using chemical cues. So carbon dioxide, for example, is a cue that they will cue on to find their host. And that will be the case, the cruising nematodes, mostly for the heterorhabditis species. And so these will be best for sedentary or slow-moving insects that can be found at various soil depths. And the steinernema more for the mobile and more at the soil surface. But then some are combining ambushing and cruising, and some will be one species of steinernema that we'll talk about on the next slide. So again, a lot about those nematodes that we don't know. We know a lot more about those kind of things with insects, but it's a lot harder to see all of that with nematodes, and there's more and more research on them. So that's exciting. So the research project that we got funded from the Wisconsin Cranberry board, and that was really asked by the board for me to assess, is those commercial nematodes. And so the rationale here is that we know that Sean Steffen has identified 2 species, Oscius honorici and heterorhabditis georgiana, that in combination as a mixture are very efficacious against multiple insects, but that includes flea beetle and other insect species. But I'll focus on flea beetle. But those are still not available commercially. And as most of you know, some growers and even consultants have been growing those, rearing those nematodes so that they can be applied commercially. But that's really a lot of work. It's really not easy, and it requires a lot of knowledge and skills and a place to do that. And so this is something that has been, I think, on the board's mind as what is the next step if we wanted to right now have a tool, especially when we're talking about losing lorsban or losing diazinon. What are the tools we're going to have? And so do we have the time to wait for those nematodes to become available commercially or to even be reared in mass by more growers? Or are there other options on those nematodes that are available commercially? And how would those fare with flea beetle control? So that's what I was asked by the board to assess. And so the project is really looking at the Steffen nematode mixture, compare that with steinernema carpocapsi, which is known as capsinum with copert. It's not the only company that will produce some of those species of nematodes, but I'll focus on the name from copert because that's the one we're testing, because copert is a collaborator and provided all the nematode species for us for free. So thank you so much for that. But this one, steinernema carpocapsi, is an ambusher, but it's potentially active at more low depth. So who knows? We'll have to see how well that does. Steinernema feltier is caparel, and that's the intermediate emboucher and cruiser. And this one also has a lower cold tolerance, which could be interesting for us in Cranberry, in Wisconsin especially. And then heterorhabditis bacteriophora, Sportinum H, is a cruising nematodes at different, can be found at different soil depths. So these 3 species could potentially offer a good alternative to other control measures for flea beetle. And also we can think about them as potential in the future being things that we combine if they are, say, an emboucher and a cruiser that are very efficacious. Maybe if we combine them, we can have higher efficacy against flea beetle. There was something else I wanted to say, but now I can't think of it. But this is what we decided that we were going to test. They are the most, I remember. Capsinum, the steinernema carpocapsi, is one that Dave Jones at Ocean Spray has been testing and has seen good results. And of course, we know that Sean Steffen has had very good results with his nematode mixture. So we have a little bit of information. We want to add some species and want to see how they're going to do. Originally, we wanted to do this project with at the field scale in half beds. We were not able to do that. So we're going to conduct a cage study for this summer on this at the recommended rate of 1.5 billion EPNs per acre for the commercial species and at a lower rate for the nematodes from Sean Steffen because of production issues and potentially because they work at those lower rates as far as we know. And we are doing 2 applications. The first one will go out next week and then the week after. And that's to coincide with what I showed you before, where the larvae were first detected in our previous research. But we're also conducting a field scale trial where we have 3 grower collaborators and we have Ocean Spray. Dave Jones has been super helpful and super kind to provide a lot of help with that. So between the grower collaborator and Ocean Spray, we're in good shape for this trial that has already started. We're doing whole bed application of the capsinum, the steinernema carpocapsi, the Steffen nematodes, and then we have a water control. These are going out via standard boom sprayers. There's not much that needs to be changed. And of course, I didn't go back and change that. We are at a regular PSI of, I think it's about 30 PSI, 20 to 30 PSI. The filters needs to be removed, but they work with standard boom sprayer. Nothing else needs to be done to adjust for the application of those nematodes, which is great news because you've heard from Sean Stephan in the beginning of his work that there might be needing boom sprayer modifications, but it turns out that it's removing the filters and having that kind of PSI works fine. So that's what we're doing. 2 applications went in, 1 last week, 1 this week, 2 more are coming, but for the same rate that I mentioned before of 1.5 million per acre, or billion, sorry, per acre. And this will be continuing on for another 2 weeks. This is done on cloudy days in the morning or in the evening, depending on the collaborator we have, but we have to avoid the sun and the UV radiation and all that. So this is important. And irrigation is following that, those applications. So very quickly to assess these, I don't know how I'm doing on time, but to assess these, we're going to be doing in the cage trial yellow sticky cards for the adult emergence, and then plant injury at the end of the trial. And then for the field scale trial in the beds, we're doing weekly sweep sets that will start in early July until late August. I want to make sure that people know with this hot weather, we know that insects are cold-blooded, so everything could be shifted, just like bloom was shifted earlier. Everything could be shifted earlier as well. So be prepared to have earlier emergence of flea beetle and other insects because of that. But this is the plan as of now, to do that in early July until late August for weekly sweep sets in the beds. And then we will characterize, and I won't go into that, but we'll characterize plant injury in the month of August with transects that we'll do, and we will do a rating within little squares, 1 by 1 foot square, of the skeletonization and how much damage we see, the browning of the uprights with different a rating scale that you have below here. And that's what I have. So again, thank you for the funding from the WCB, the nematodes that are donated by Copert and the Stephan Lab. And of course, we couldn't do that without all the wonderful grower collaborators. Dave Jones, really thank you so much, and Ocean Spray.
Fantastic, thank you. I had one quick question way back in the far past of your presentation about Ben Jaffe's data on when you were able to collect eggs versus larvae. Do you recall what county or what part of the state that was done in? Just because I know it'll shift mid-state versus the northern half.
Yeah, that's a good question. What I would say, it was a long time ago, but usually we try to do them as close as possible, not as possible, but closer to Madison, because we come back weekly and it's a lot of work, but it was done at multiple farms. It wasn't done at just one farm. So there was a lot of...
Oh, okay.
Oh, actually, I might take that back. I don't recall. So I shouldn't say that. I would have to go back and check how many farms we did that at. It was a ton of soil cores. So I don't remember. I think they were done on multiple farms, but I could be wrong. I'm mixing all my trials.
But I guess as usual, as when you as a grower are listening to this presentation and seeing a date, remember to mentally shift to the correct, date range for your location. That was a lot of really cool information. I'm looking forward to seeing the cage study outcomes. Any other questions in the chat, or unmute yourself and ask? Otherwise, we will pass it to Olivia. Thank you so much, Christelle, for the flea beetle background, which is always good to get, and then the exciting new work on the potential new ways that we can get them taken care of.
Thank you. Yeah,
it's plenty to keep us busy. Any other questions, either in the chat or elsewhere? I just want to thank our presenters again, and wish everyone a productive rest of the Friday afternoon, a productive and pleasant blossom season.
Thank you.